for being here. And I just wanted to present Dr. Flagel really briefly because I always do this. So, and this comes from her website, just for my students to know. Dr. Flagel is Associate Professor of Psychiatry and Molecular and Behavioral Neuroscience Institute. And she's an alumni of the Neuroscience Graduate Program, and now she goes back to her alma mater as Associate Professor. And she's Associate Member of the American College of Neuropharmacology and the Kavli National Academy of Science Fellow. Her research focuses on understanding the behavioral and neurobiological factors that contribute to individual differences in reward learning and susceptibility to addiction. And she's specifically interested in the psychological mechanisms that undermine and influence appetitive Pavlovian learning and the neurocircuitry involved in these processes. And most recently, she was awarded the Society for Biological Psychiatry, the Bennett Basic Research Award, for her distinguished career in the field of behavioral neuroscience. And another thing, I don't know, I would just mention is that Dr. Flagel was an alum or a mentee of Dr. Huda Akil as well. And there's a connection with me, Shelly, because one of our students, Javier, I don't know, maybe he was.
Oh, Javier?
Yes. Well,
yeah, I'm good friends with him.
You remember? Okay.
Oh, yeah, of course. No, we still keep in touch.
Okay, well, Javier was my student in my lab.
Oh, I didn't realize that. That's awesome.
Yes. So you have to mention to him that Dr. Maldonado said hi.
I will.
Because, yeah, so before going to, University of Michigan, then he was here in my lab. So I just
That's awesome. Yeah. We teased because his little girl and my son were born at the same time. So we said that they went on their first date together when they were like a month old.
So wonderful that, the disease, neuroscience is a very close community. So
yeah, that's so funny.
Yes. So without further ado, thank you, Dr. Flagel, for being here. And please, you can start with your short summary.
Yeah, no, so it's really my pleasure to be here. As I mentioned before, this is the first time I've done Google Meet's presentation, and it seems like I won't be able to see you or won't be used to interacting with you, so please feel free to chime up if you have a question during my presentation. And the plan is for me to give a relatively brief, probably 20 or 30-minute presentation on my research, and I think it's representative of at least a subset of research that goes on at the University of Michigan. And then we'll open it up to general questions about my science or our science, because Amanda, who you'll be talking to later, is in my lab, and Christina's a collaborator. And then up into our program in general. So thank you again for having me. Let's see if I can figure out again how to present. Okay. So hopefully you can all see this. What I'm interested in is studying the neurobiology underlying individual differences in susceptibility to psychopathology. And what I hope to convey to you today is how we can go about that by studying the way individuals respond to cues in their environment. Before I get there, I just want to tell you a little bit about my path and my route. And so I actually have been at the University of Michigan, not just for graduate school, but I came here for undergrad. And when I came here, my whole life, I thought I wanted to be a veterinarian, and it was just kind of without question. I loved animals, I loved studying their behavior, and really what I knew that fell into that realm in terms of professions is being a veterinarian. Once I got here, though, my sophomore year, I got introduced to the idea of research. And so I started doing research, and at that time, I was kind of wavering, and I had, veterinary medicine, you have to take the same pre-med courses as those going to medical school. And so by that time, by my sophomore year, I was becoming a bit disenchanted with that route in terms of undergraduate courses and the competitiveness and so on. And I got introduced to the idea that I could participate in research as an undergraduate. The whole idea of research, or even the possibility of becoming a professor or dedicating my career and life to research, was not something that had been on my radar before I came to undergrad. And at the time, I was starting to consider whether or not I wanted to go into psychology, and in particular, clinical psychology. And so when I was looking for a research position, I was very interested in obtaining a clinical position, so that is working with human subjects. And sure enough, I was lucky and able to do that. And I ended up on a project where I was working with individuals who had gone to Vietnam, and some of them, and most of them had been exposed to combat, and some of them developed post-traumatic stress disorder, and some of them didn't. And so my job as a research assistant was to play combat noise to them, and then they would eventually go to the brain scanner, and we'd see how that elicited different brain responses. And that really just hit home this notion of individual differences and the question of why is it that some individuals are more susceptible to mental illness than others. And that, in combination with my classes, my abnormal psychology classes, my biology classes, and others, just really made that question stick with me throughout my career. So after my undergrad degree, I ended up actually then, for the second part of my undergraduate career, I was going to do an honors thesis. And my mentor at the time told me there was no way I could do an honors thesis working with human subjects. And so he convinced me to go into the lab and try to create an animal model of what I was seeing essentially in the human subjects. And so that's when I got introduced to animal models, and I did an animal model essentially trying to capture aspects of post-traumatic stress disorder. And so then I fell in love with the lab. And when it came time to applying to graduate school, I was still a bit torn because I still had this image of what kind of a mad scientist looked like or what a neuroscientist looked like. And most of them were male, and most of them were kind of locked in basements and rarely interacted with other people. And so I really questioned whether or not that's what I wanted to do with my career. So when I applied to graduate school, I actually applied to clinical psychology programs, to behavioral neuroscience programs, to biopsychology programs, and to neuroscience programs. And I ended up choosing neuroscience at the University of Michigan because I knew out of all my interests, so I narrowed down my interest to maybe 3 to 5 topics. And out of all of those, I knew that Michigan was big enough and that there were enough great people here that no matter what I decided to do, I'd be able to find an expert and have the resources to do it. And the other reason I chose it, and we can talk about this more later, is because this is true today too, and another reason I encourage people to choose our program is because we allow you to do rotations, and in fact we encourage rotations. So when you come to the University of Michigan as a neuroscience graduate student, you should do at least 2, but ideally 3 rotations before you commit to a thesis lab. And so that really gave me the option of exploring my vast interests that were very diverse before I decided in what lab to do my thesis work. So I ended up coming to Michigan, got my PhD in neuroscience, worked under Delia Vasquez and Terry Robinson to study the effects of early life stress on drug-taking behavior using an animal model. And so that's when I started to become interested in addiction. And then finally, as my postdoc, I joined the lab of Dr. Huda Akil, also at the University of Michigan. And she is an expert in really the neurobiology of stress and emotionality, and she was already studying individual differences. So it was a great way for me to kind of enhance my molecular skills while really diving into this field of individual differences. And then finally, I joined the Faculty of Psychiatry almost a decade ago now. So I've had my lab for almost 10 years. So today what I want to talk to you about is how it is that I get back to this question that really piqued my interest in undergrad. And that is, why do individuals differ in their susceptibility to psychiatric disorders? And I'm going to talk to you about it with respect to how individuals respond to cues in their environment, because I think that this is actually relevant to the development of psychopathology across psychiatric disorders. So that is, I think that there's shared symptomatology across psychiatric disorders. And by studying the mechanisms that I'm going to describe to you today, I think that we're starting to uncover what those factors are that contribute to this enhanced susceptibility. So if you were to come to Michigan, to Ann Arbor, where the University of Michigan is, I would bet that it wouldn't take you long to recognize what these cues and symbols mean. So we have a few fantastic coffee shops at the University of Michigan, or in Ann Arbor, one of which is Mighty Good Coffee. We have an amazing ice cream place, Blank Slate, and we have some pretty good bars, as you may imagine, like Hopcat. And so these cues or symbols that are outside of these establishments, once you experience what's inside, will come to have some meaning for you. And again, this is based pretty much on your learned associations with these establishments. For me, me, my particular weakness is Starbucks. So even though I recognize that we have much better coffee places than Ann Arbor, there's a Starbucks sign that's close to the psychology building on campus. So when I come from the medical campus to the psychology building, I see that Starbucks sign, I'm drawn to it, I'm attracted to it. I go into the store and I purchase my cup of coffee, regardless of the time of day, regardless of how much coffee I've already had that day, or how much money I've spent on coffee that week. So I like to think of this as an example of how cues or stimuli in the environment can regulate and guide our everyday motivated behaviors. But such stimuli associated with rewards can also lead to maladaptive or psychopathological behaviors. So one of the best examples of this is if you consider an individual with addiction walking down the street and they come across the sight of this used needle. The sight of that needle alone will trigger this irresistible urge and desire, leading to drug seeking and drug taking behavior or relapse. So how is it that this happens? How is it that cues in the environment attain the ability to control behavior? This has to do, again, based on your prior experience, but based on that, you attribute some sort of value to that cue. And that determines the extent to which it controls behavior. So a cue attributed with what we call predictive value simply signals the availability of reward. A cue attributed with what we call incentive value can trigger abnormal motivational states and lead to maladaptive behavior, as is the case with the individual with addiction that I showed you a minute ago. We actually know, based on, I'm sure you've all heard of Pavlov's dogs and Pavlov's classical conditioning studies, and we know actually based on his book that was published in the late 1930s that there was great individual variation in how dogs would respond to these cues in the environment that were paired with reward. So only some dogs would show this, what he called sort of great hyperexcitability, whereas others would be much more inhibited and show less reactivity to cues that predicted the reward. And so we know, based on these studies and a number that have followed up, looking more at the psychological phenomena, that individuals do vary in the extent to which they attribute value to a reward cue. And we know that cues that are predictors, that is, cues that attain predictive value, don't necessarily act as incentive stimuli. That is, only some individuals attribute both incentive and predictive value to a cue. So only for some individuals do the cues transform into incentive motivational stimuli. So how do we study this in the laboratory? Again, we use a classic Pavlovian conditioning paradigm. So that means a lever comes out into an illuminated chamber, and that lever is paired with a food reward. So in this case, it's pairings of lever, food, and the lever therefore becomes a conditioned stimulus, the predictor of the food reward. The food reward is the unconditioned stimulus, or the US. And so we have these lever food pairings, and there's no response required for the food to be delivered. And that's what I mean by classic Pavlovian conditioning. The food will always follow lever presentation. Yet we see that these distinct conditioned responses emerge. So on one extreme, when that lever is presented, these individuals on one extreme, what we call sign trackers, will approach and interact with the cue and manipulate it, almost as if it were the reward itself. And only after the cue is retracted, the lever is retracted, do they go to the food cup to get the food reward. On the other extreme, we have what we call goal trackers. So they see that cue for what it is. It's merely a predictor. It's predicting the availability of food reward. So they see the cue and they simply go and they wait for the food to be delivered. And so we can quantify these behaviors and look at things like when that cue is presented, what are they doing? And you can see here that sign trackers are interacting with the lever, increased number of lever contacts over sessions, increased probability to contact the lever, and a decreased latency to contact the lever. Whereas on the other extreme, goal trackers in the orange are showing increased number of head entries into the food cup when levers presented, increased probability to enter the food magazine, and a decreased latency. They do so more and more quickly over time. What's really important here again is that these responses are not required. The food's always going to be delivered and they all consume all of the food pellets, but yet these distinct conditioned responses emerge. And so you can see here that, again, this is just highlighting the individual differences. So each line is an individual rat, and at once an extreme of the population develops sign tracking behavior and the other extreme goal tracking behavior. And then finally, you can see here that we've been doing this for quite a number of years. We've collected a large database from all of our collaborators at the University of Michigan. So there's a number of us in the behavioral neuroscience under that rubric that that study these types of behaviors. And so we have a database of over 6,000 rats. And when we look at that database, you can see that about a third of the population are sign trackers, about a third of the population are goal trackers, and about a third are what we call intermediate responders. So you can see this guy here, he's holding onto the lever, but he's also putting his head into the food cup. So intermediate responders will vacillate between the two responses or show them both at the same time. So hopefully what you've gotten out of these first few minutes is that cues can be predictors and elicit a conditional response. And it does so for both sign and goal trackers in this case. For sign trackers, however, because the cue is attractive to them, the cue is also what we call an incentive stimulus. So it's transformed into a motivational magnet. So Kent Berridge and Terry Robinson in the psychology department here have been studying this concept of incentive salience for years. And Kent's termed this type of cue as a motivational magnet. That is, it itself becomes attractive and desirable. And we know that it obtains condition reinforcing properties. So one of the ways we can assess how reinforcing it is to these sign trackers is to ask, will they work for presentation of that cue in the absence of reward? And they do so, and they do so to a greater extent than goal trackers or intermediate responders. So why do we care? So this is really complex psychological phenomena that I've described to you so far. And now I want to try to convey to you why we care. So one is, by uncovering these psychological phenomena and capturing the behaviors that do so, we can now study the underlying neurobiology. And that's important because to date, or until we started using this animal model, these properties were confounded. And so we would just kind of lump things into motivation and say, oh, we're studying the neurobiology underlying motivation. But that didn't get us too far yet. And so we think that by being able to parse the predictive versus incentive learning processes and uncovering the associated neurobiology, then we'll start to really understand, for example, why cues in the environment or drug-associated cues attain inordinate control over behavior and lead to increased relapse propensity. So what I'm going to do now is walk you through a few examples of how we've utilized this model to study the underlying neurobiology. And one of the first things we wanted to do was to assess the role of dopamine in these processes. And that is because, as I'm sure you know from pop culture literature, people often say dopamine is sort of the pleasure molecule. It has to do with reward. Well, if you're in the neuroscience field of reward, learning, and motivation, you know that dopamine is much more complex. It has a much more complex role than that. And so we wanted to be able to say, to parse the role of dopamine in reward learning and just determine whether or not it's involved in predictive versus incentive learning processes. To do this, we paired up with Paul Phillips and Jeremy Clark at the University of Washington, and more recently, Amanda Iglesias, who you will hear from later as a graduate student in my lab, has done some great work with Optogenetics to take this work, our findings from several years ago, one step further. So some of you may have heard of the prediction error theory. This is a theory that actually, when I was in graduate school, I remember when this paper came out and it was just kind of a game changer and people were in awe, because now all of a sudden, what it showed was that dopamine has more to do with the anticipation of responses rather than responses related to reward seeking specifically. And so these are just showing you traces of signaling of dopamine neurons. And up here, what it's showing is that, if you just give, in this case, it was a monkey, but now they've shown it in rodents as well as humans. But in this case, if you give a monkey reward, you're going to get an increase in dopamine neuron activity. But then if you pair that reward so that now every time before that reward is presented, you have a stimulus or a cue presented. So it's cue reward pairings again. Now that dopamine response shifts from the reward to the cue itself. So you see this classic shift that's now described as a classic shift in dopamine from the reward to the cue. And that's thought to signal the anticipatory response or the predictive value of the cue. So then we asked, well, if dopamine truly is encoding the predictive value of reward cues, then we should see the same pattern of dopamine responding in both sign and goal trackers, because a cue is a predictor for both. It elicits a conditional response and is a predictor for both, and only for sign trackers does it have that added incentive value. When we did this, we used fast-scan cyclic voltammetry to get sub-second dopamine release in the nucleus accumbens. And this is just a fancy voltammogram. This is showing you the traces per session, representative traces. And you can see that for sign trackers, you initially have this response to the reward. And by the end of the training, you have this response shift from the reward to the cue itself. In goal trackers, however, the shift is absent. You're not seeing a clear shift from the reward to the cue in goal trackers. So what this told us then was that dopamine isn't necessarily encoding the predictive value, but rather it seems to be encoding the incentive value. Let me show you some examples of videos. This is showing you the dopamine trace in the bottom in yellow. Here you're going to see the cue come out over here. This is a sign tracker. When that cue comes out, you see this big increase in dopamine. In response to the cue itself, it retracts, and then you don't see much of a response in response to the reward. In contrast, you have this goal tracker. In this case, the cue is going to come out over here. The cue's out, it orients to the cue, it looks, but its big dopamine response doesn't come until after the cue retracts, it gets the food reward, and now you get dopamine response in response to the food. And so to me, that was pretty remarkable. These animals are exposed to the exact same training, the exact same paradigm, but their dopamine is responding in different ways. And what we had concluded based on these findings was that in sign trackers, the dopamine is encoding the incentive value of the reward cue. Well, next, we turn to classic pharmacology. If that's the case and dopamine's encoding the incentive value, then we should be able to block dopamine and block sign tracking behavior, right? So what we did is we took animals, these are bred high responder rats from Huda Akil's lab that we knew they're bred based on locomotor response to novelty, but we knew that they would also be sign trackers. So that is, bred high responders were always sign trackers. So these bred animals that we knew were going to be sign trackers here in the open circles develop a sign tracking response. But if we give them flupentixol, dopamine antagonists systemically, prior to training, you can see that you don't see the development of a sign tracking response. So that was what we had expected, right? But if you give goal trackers, so these are bred low responder rats that we knew were going to be goal trackers, you can see their trajectory and goal tracking behavior. If we give them flupentixol, we saw an attenuation of the goal tracking response. So that was surprising to us because we thought, based on the prior studies I showed you with voltammetry, that by blocking dopamine receptors, we would block just sign tracking behavior. But the truth came out after we then took these animals off of the drug. We exposed them to another session to see what their behavior looked like. Here you can see in the sign trackers that you take them off the drug and they still show a great attenuation of the sign tracking response. So they show no evidence of having learned that cue reward association or having attributed incentive value to that cue. In contrast, if you look at the goal trackers, you can see that once you remove the drug, you take away the dopamine receptor antagonist. Now you can see that there's actually no evidence that they had ever received the drug in the first place. So they were fully capable of attributing predictive value to the cue during the learning paradigm. It just didn't come across until after you removed the dopamine antagonist. So that was all done with systemic administration of dopamine receptor antagonists. And then when Ben Saunders and Terry Robinson's lab did central administration into the nucleus accumbens, you can see that you still see an attenuation of sign tracking response without affecting the goal tracking response. So next, we wanted to turn to more recent techniques like optogenetics, and this is work done by Amanda Iglesias. And we wanted to now ask, well, what happens if we block that peak increase in dopamine, that little yellow peak that I showed you in the video? What happens if we block that during cue presentation? So to do this, we turned to transgenic rats, in this case, tyrosine hydroxylase Cree rats, which would allow us to specifically manipulate dopamine neurons. And you can see here that by putting in certain viruses and using a laser light, we'd be able to selectively inhibit dopamine neuron activity. And that's what we're doing here. And we decided to do so when that cue is presented. Again, with the notion that we're blocking that peak dopamine response that I showed you in yellow, we're blocking that and we want to see what happens to behavior. First, before I show you the results, I want to take a special note to show you that we discovered that in our hands, these TH3 transgenic rats happen to almost all be sign trackers. So they're skewed towards sign tracking. In our case, that's actually an advantage because now we can ask in rats that are prone to sign tracking, if we inhibit dopamine, do we block sign tracking? But it's a note of caution because as we get on board with these novel technologies and these transgenic rats and mice that are available, we need to really pay close attention to behavior so we're sure that we know what we're assessing. And so this is showing you what happens. This is a control animal, so meaning they don't have virus. The laser light comes on, the lever comes out, and they sign track as expected. In this case, when the laser light comes on, we're actually inhibiting dopamine neurons. You can see the lever's out here, and this animal doesn't care much because his dopamine neurons are being inhibited. So it's not till the lever retracts that it then goes and gets a food pellet, but it's never approaching or sign tracking towards the lever when its dopamine neurons are being inhibited. And so we can quantify this, and so the way we did this is we inhibited dopamine neurons during cue presentation during those first 3 sessions, and you can see here that we're attenuating sign tracking, and we take them laser off, and they eventually learn the sign tracking response. Interestingly, we're not seeing any difference in goal tracking behavior, perhaps because these animals are skewed towards sign tracking in the first place. And then this is just another way to quantify the difference between sign and goal tracking. And you can see laser on, and we have a cluster down here more towards goal tracking, but you take the laser off, and now all of a sudden they start to sign track. So as an intern summary, hopefully what I've convinced you of is that shift in dopamine from the reward to the cue is actually encoding the incentive value of reward cues and not the predictive value as was once thought. And dopamine also, using pharmacology, we show that dopamine is critical for the attribution of incentive value to reward cues and the development of sign tracking behavior, but not goal tracking behavior. And other people had shown that dopamine directly into the nucleus accumbens core is critical for the expression of sign tracking behavior. More recent work by Amanda has shown using optogenetics that indeed, if you inhibit dopamine during that cue presentation, you attenuate the development of sign tracking behavior. So again, I hope that this was just a good example of how we can use different techniques ranging from classically and cyclic voltammetry, microdialysis types of techniques, to pharmacology, to more recent techniques like optogenetics, to really probe the role, on top of complex psychological phenomena, to really probe the role of these, the underlying brain mechanisms. And so now, more recently, what we've done is we've turned to not just specific molecules, but more on a circuit level. So we're starting to use techniques to probe neural circuits that encode the predictive versus incentive value of reward cues. I'm not going to show you much data, but I just want to give you a sense of where we're going with this. So what we've done is if we put these rats into the chamber and we train them to sign and establish that they're sign or goal trackers, and then present them with the cue. In this case, you consider it the Starbucks sign, and then we look at cue-induced brain activity patterns. What we've seen is that very distinct patterns emerge between sign and goal trackers. So these are what I call my correlation wheels, looking at cue-induced correlated CFOS activity between brain regions within sign trackers or within goal trackers. What's important here is that if you look, you can see there's no engagement of the cortex in sign trackers with other subcortical structures. Whereas in goal trackers, you have this cortical engagement of subcortical structures. What this means is, I think, is that goal trackers are using more of this kind of top-down cognitive learning strategy, relying on executive function and relying on this top-down inhibitory control to regulate their behavior. Whereas sign trackers are lacking that cortical top-down control and otherwise relying on subcortical, just sort of purely motivationally driven processes. And we've done a number of studies to show that this is the case. So a few that I'll highlight. More recently, we've used a chemogenetic approach to selectively look at the cortico-thalamic circuit from the prelimbic cortex to the PVT. And that data does support our hypothesis that this top-down corticothalamic mechanism is actually kind of putting the brakes on these subcortical processes that are encoding the incentive value. So if you have this corticothalamic system turned on and ramp up, you're going to be able to inhibit any subcortical processes encoding the incentive value. Christina was involved in a more recent study where we're starting to look at what are those subcortical mechanisms that are encoding the incentive value. So we've lesioned the lateral hypothalamus where orexin originates, and we've shown that you can lesion that and you can block the encoding of incentive value. You can also specifically inhibit orexin in the PVT and block the incentive value or block sign tracking behavior. More recently, Amanda, in addition to her looking at the dopaminergic projections, she's been specifically targeting using a chemogenetic approach, lateral hypothalamus to paraventricular nucleus of the thalamus projections, using chemogenetics to ask, well, if we inhibit these subcortical processes that are presumably encoding the incentive motivational value, are we inhibiting sign tracking behavior, and vice versa with stimulation? So we're starting to get a really good idea of the circuitry underlying these distinct forms of learning. So hopefully what I've convinced you of is that if you pay close attention to behavior, and in particular individual differences, you can start to learn a lot about the brain mechanisms underlying very distinct psychological phenomena that are relevant to the development of psychopathology. But we also, again, back to this question of why do we really care? And well, again, I think that there is this tight relationship between that propensity towards incentive learning and the development of psychopathology. And so we wanted to further investigate what that relationship is. And so we and others, actually almost everybody on this list I'll show you is at the University of Michigan, all involved in the neuroscience graduate program. Well, we've looked at things like addiction-related behaviors, and we know that sign trackers show enhanced response to cocaine and enhanced propensity for cocaine for reinstatement or relapse to cocaine-seeking behavior. We also know that sign trackers are more impulsive than goal trackers. They have attentional deficits akin to ADHD. They have increased propensity for falls. So attentional deficits and the propensity for falls is all work by Martin Sarter. So Martin is interested in both attentional cholinergic and cortical attentional processing, but also models of Parkinson's disease. And so we think actually that this may be relevant to Parkinson's disease in some way. And then work from Jonathan Morrow, which is where Christina's doing her dissertation work, looking at fear response. And so sign trackers have an enhanced or exaggerated fear response to aversive cues. And so Dr. Morrow's lab has this model of sort of comorbidity between increased propensity for addiction and post-traumatic stress disorder. And so we think then, given all of these behavioral differences that we've seen, that we're capturing this, what I call neurobehavioral endophenotype of relevance to psychopathology. And again, this is just showing you sort of, it may look like a complicated subway map, but it's a blueprint of work from our groups as well as a number of others over the last decade or so, showing the different brain regions and brain mechanisms and circuitry that we think are involved in these processes. And I'm going to, again, pitch the notion that it's an imbalance between these top-down sort of executive function, inhibitory control processes, and these subcortical arousal and motivational systems. And when an imbalance occurs, and these aren't properly balanced, presumably by like midline thalamic structures, when that's out of balance, then you get these extreme behaviors that are characteristic of a number of psychopathologies. Of course, to know whether or not the propensity to sign track is truly relevant to the development of psychopathology, we need to turn to humans, and that's exactly what we've done in recent years. So originally I paired up with Michelle Joyner and Ashley Gerhardt in the psychology department here at the University of Michigan. And I wanted to collaborate with them because they were the only people at the time working with human subjects that were willing to let us kind of develop this paradigm based truly on what I'm doing, what we had been doing in rodents. And so you can see here that these kids, and this group of kids, the first group we did were 5 to 7-year-olds, are truly exposed to a Lego box where an illuminated lever comes out just as it did in the rat chamber. It retracts and they receive candy in the food cup. And so I'm going to show you some examples of what these behaviors look like. So this little boy, you can see he's quite exploratory and interested and kind of cognitive and cerebral, trying to figure out how things work. The lever comes out. He actually shakes his hand in his head, engages that cortex, doesn't approach the lever, but goes and waits for the food to be delivered. In contrast, this individual, the lever is going to come out here. The lever comes out and you can see they're immediately attracted to it. They interact with it, they're pressing it, and so they're clearly a sign tracker. And so we're starting to see these extreme behaviors emerge using the human paradigm as well. And we have a ways to go and a number of studies to do, but I just want to show you some of the parallels that we've drawn here, where you can see a sign tracking human child and sign tracking rat, an intermediate responder, and then a goal tracker. And so, like I said, we have a ways to go, but I'm really excited about being able to translate our work to the human population with the hope of characterizing them at a young age and knowing, being able to predict, capturing something in our behavioral output measure that will predict whether or not they develop psychopathology, in the hopes of developing novel treatments for the treatment of psychopathology. So just in a very general summary, and admittedly, this is a very biased summary towards behavioral neuroscience, and I encourage you to ask questions to get a good glimpse of what else we do at the University of Michigan. But what I want to pitch to you is that by studying adaptive and maladaptive behaviors in animals, we'll be able to better understand the development of psychopathology in humans. And really, I think that behavior can serve as the common language between species. And I think by studying it, we'll be able to really better understand the brain. And perhaps most importantly, and for you guys, as you embark on these careers in neuroscience research, to be able to embrace the complexity of the brain and behavior and recognize the need for team science. So with the advent of all these new technologies, it's almost impossible to be able to do everything yourself or everything within one lab. And so team science and collaborative science is going to be critical for making real scientific advancements that will help humanity. And I'm also biased, but I think the University of Michigan is a great place to do that, given our very sort of collegial and collaborative style and our vast and large program that gives us access to many resources and colleagues. Let me stop sharing. Okay. All right, I can see you all now.
Wonderful. So, okay. I love everything what you said because it's close to my scientific research, of course, on
oh, that's good.
and context learning. Questions for Dr. Flagel? I know they are. Any questions? You can unmute yourself if you want. I think it's better.
Okay, I can go first. Hi, Dr. Flagel. My name is Viviana, and I want to ask you, so how long does it take for you to notice these phenotypes? Is it often from the first session, or does it develop throughout time?
Yeah, that's a really great question. So typically, we screen animals based on 5 sessions. And the reason that is, so each session has 25 trials. By session 3, like 75 trials or so, you can start to clearly see the phenotypes emerge, but then we just want to make sure they're stable, and so we typically go out to 5 sessions. But your question is fantastic, because ideally what I want to do is I know that there has to be something on day 1 that really determines whether or not they're going to be assigned or goal tracker. So I've worked with a number of computational neuroscientists and bioinformatics people and given them our data of the 6,000 rats, and nobody can give me anything that shows that. But what I want to start doing is using things like deep lab cut, so fancier technology to analyze our behavioral videos. It's fancy, but it's also open source and free to be able to really analyze our behavior in greater detail and to maybe then be able to identify something on that day 1 that will allow us to predict. And then the hope is to apply similar technology to humans to do the same.
Thank you.
Yeah.
Yeah, Viviana is very interested in behavioral work at very much. So her background is passive avoidance methods. She works with Gregory Quirk, and I know.
Yeah.
So she is.
No, I know Greg well.
She's looking for a laboratory that really excites her. So we'll see what she chooses later. But yes, wonderful.
Awesome.
As well. Any other questions? Vamos, questions?
I can keep asking. I have another one.
Okay, Viviana, go ahead.
Okay, I know that you mentioned that someone else was studying the comorbidity of sign trackers and also seeing these behaviors in a fear conditioning, fear extension kind of paradigm. So I don't know if you can maybe talk to me about that. I don't know, maybe sign trackers tend to be less resilient than goal trackers. I don't know if that's the case.
I'm going to turn it over to the more of an expert on this call. Christina, would you like to take that one?
Sure. So basically, when you subject sign and goal trackers to cue fear conditioning paradigms, so where you expose them to, a cue that predicts a shock, in this case, we use a tone. What we see is that when we do a cue test, so after you've done the fear conditioning and now you expose them to a different context, so you show them the cue, sign trackers tend to react a lot more, have hyper reactivity to the cue compared to goal trackers. And what we think is that somehow, this sensitivity that sign trackers have to cues is regardless of the emotional valence of the cue. So whether it is aversive or repetitive, it just seems that this seems to be the pattern. And, this is very important for comorbidity because when you look at the rates of people with PTSD that also have addiction is a lot higher than in the general population. So there seems to be a very strong link between these two disorders. But yeah, we're trying to, in John's lab, Dr. Jonathan Morrow's lab, we're trying to adjust the aversive task because a cue is not necessarily the same as the lever, like at least the freezing behavior that we study as a response to the cue is not the same as the approach behavior that you see with the lever. So it's interesting that you're studying avoidance because what we're trying to do is do a task where the lever is going to be the predictor of the shock, and then there'll be a platform below the lever. And so that way, if sign trackers really have a strong aversive response to this cue, then they'll sacrifice the safety of the platform in order to stay away from the lever. And so that'll be a more direct measure of kind of like a negative sign tracking response compared to just, looking at freezing that is not exactly the same as approach. So yeah.
That's pretty cool. Do you also are you thinking about looking at extinction of this avoidance behavior too?
Yes. So yeah, that would be, part of the experiment, just to look at whether it's just any different between sign trackers and goal trackers, especially because we already know that there's extinction differences for repetitive behavior between sign trackers and goal trackers, where sign trackers seem to just require more extinction trials. So probably we would see a similar pattern with fear conditioning.
Okay, great, thank you.
Yeah.
So I had a question.
See?
Do you see the same behaviors in mice?
Yeah, that's a good question. So yes and no. Yes, people have. There's a few papers that have done it almost identical to the way we have, and they've reported individual differences. There's a great paper by Alyssa Chesler, who I think looked at 12 different strains of mice and showed that it's highly dependent on the strain, which is not too surprising because we actually know that that's the case even in rats. And we know that there's great variability depending on the vendor we get the rats from and even the barrier room. So all of this pointing to a genetic basis. And I gave you the example of our TH Cree rats being all sign trackers. So the variability you get, I think, is going to be highly dependent on the strain that you're using. And there's a few papers that have highlighted that, but then there's other papers that have tweaked the paradigm a little bit to be able to better get individual differences in mice. So I'm happy if you want to try to set it up. I'm happy to talk to you or put you in touch with people that have done it. You just have to tweak a few things. So it's a little bit different.
Thank you.
Yeah.
And what about gender differences?
Yeah, great question. And I should have put that slide in. So if you look at raw values of sign tracking or goal tracking, we don't see much of a difference. But if you take the population as a whole, females are skewed more towards sign trackers. Yeah. So we've shown that repeatedly, and it always surprises me that the difference is being carried by the intermediate population, which tend to be a little bit more sign tracky. But if you look at the raw values of the extremes, you don't see differences between males and females.
But it's interesting because in the human data, at least the video you showed, that the girls
Yes, but that's not, so that was the first thing. So I have 2 boys, and the first thing I looked at, I was like, there's got to be sex differences because I'm always jealous of people with little girls. But there wasn't, that was obvious in the 5 to 7-year-olds. But we're now looking at the 9 to 13-year-olds and then even older. So I'm thinking that with increased sample size, we've got to, I'm sure there'll be sex differences. I'm sure there will be.
Very interesting. Very interesting. Any other questions? You can unmute yourself.
And of course, I'm happy to respond to email at any time. I can put my email in the chat.
Of course. Okay, well, I don't know if you wanted to start the part of the recruitment, if you wanted to show some slides or just start talking with the students. I see Tivo join us, so hola Tivo. So Tivo is a former Neuro-ID student and a proud alumni of our institution, and now he's a graduate student with you. So we also are very proud of Tivo.
So are we. So yeah, actually you have 3 superstars here to nicely represent us in terms of, you know, getting their independent funding and just being great students and community members in general. So maybe we'll start with introductions, and Audrey Seashultz, interim director of the program, is also here to join us. And I'll let you guys do introductions while I pull up the other slides if we feel like we want to go that route. So go ahead.
Okay, I can go first. I'm Audrey Seashultz. I'm, as Shelly said, the current interim co-director of the neuroscience graduate program, and I'm really excited to talk to you and try to answer any questions you may have about the program. I study stress in my lab. I use mice, and we study stress and its association with anxiety, depression, and alcohol disorders.
Can you guys hear me? Yeah. Okay. Hi, everyone. I'm Amanda. I don't have to cover my research because Shelly pretty much covered all the bases. But I guess I could start with a little bit of my trajectory. So I'm from Southern California, and I did my undergrad at Cal State Fullerton. It's a really great school, but it's kind of a commuter school, so there's no like community. People just get their degrees and go out. I initially wanted to be like a clinician or something, but I took a biopsychology class and it really intrigued me, so I decided to pursue research just for fun and just have something on my CV when I applied to be like a psychiatrist. But then I fell in love with it, and I ended up joining the MARC program, which is similar to the BP Endure program, right? Yeah. And so they encourage you to go to a summer program, and I did mine at the University of Michigan in a lab in biopsychology. And they studied sign and goal tracking, and that was my first like taste of individual differences and the world of behavioral neuroscience. And I kind of fell in love with it and like needed to know more because it was a short 3-month program and I didn't really learn or do much. So I applied to University of Michigan, got in, and rotated in a few behavioral neuroscience labs, but ended up being the Flagel lab, and I'm completely happy. So wherever you end up in Michigan, if you come to the neuroscience program, I think there's a lot of PIs that are great and supportive and will help you grow. So if anybody has any questions about route to graduate school or NGP in general, you can feel free to email me. I can put my email in the chat as well. But yeah, that's pretty much it. And I study individual differences, as Shelly covered.
I guess I can go now. So my name is Cristina, and I was actually in the MARC program as well at Rio Piedras for 2 years, so from 2014 to 2016. And I was doing my undergrad research with Dr. Carlos Jimenez at the Medical Sciences campus. And I actually, came straight from undergrad to Michigan, mainly because of the recruitment efforts from PIBS at the UP. Like, I think they went twice in the time that I was in MARC. And so I started really getting interested in looking at what labs were in Michigan. And I came from an addiction research background. So I quickly realized that Michigan was an amazing place to study addiction and just, motivation and reward learning, but a lot of other things as well. We're all kind of biased here, but well, I guess Toefl is doing different stuff. But anyways, I came to Michigan, and it's been really great so far. I think Michigan really gives you the tools to develop into a not just like an independent scientist, but I think it allows also like a level of creativity, just because of the immense collaboration network within the university, and then just the access to a lot of techniques and resources. And I rotated first with Dr. Morrow, Jonathan Morrow, and then with Dr. Murphy, or Jeff Murphy. And I just decided to do a co-mentorship with the two, just because I was really interested in doing electrophysiology and the sign and goal tracker model. So again, it's the same background for the research that Dr. Flagel explained. And Dr. Murphy is an expert in electrophysiology. And so we just decided to do a big collaboration. So yeah, I've been doing a lot of electrophysiology and sign and goal trackers, and then also doing chemogenetic manipulation of specific circuitry to also kind of understand where these individual differences are coming from. So I've really been able to, branch out with my thesis work and just learn a lot of different, kind of like target this model at a lot of different levels that I know it's going to help me eventually. So yeah, if you have any questions about anything, just feel free to reach out or ask questions now. I'll also put my email in the chat.
Okay, so my name is Thibaut. Like La Doctora said, UPRRP, 2016. It's been a while. So I did my undergrad there. I was part of the Neuro-ID program. At first, I joined the Neuro-ID program because I just needed some financial help, and I wanted to get into med school. Sorry, I'm guilty, very bad. But I fell in love with it. It was really a fun environment to be in La Doctora's lab, where undergrads had so much freedom to explore and to learn. I really, really, really appreciated that. But afterwards, I still was thinking maybe I want medicine. So I did a postbac. And so I did a postbac at the Medical University of South Carolina with Peter Kalibas, because I wanted to do some shadowing, and I wanted to know. I didn't want any regrets. But during the whole year, the experience was that science was more for me and not medicine. And as I kept going through the science, I liked it more and more. And so afterwards, I applied to University of Michigan. And particularly, I decided to go to University of Michigan because I really like the collaborations. Collaborations when I was an undergrad was key for me. Some equipment like Westerns and whatnot were taught to me by other labs, and that was key for my for the paper that I managed to get out of there. And so I was looking for a place where I wouldn't be tied to just the lab funding or just the lab environment or what they did or their specific techniques, etc. I wanted a place where I could just learn whatever I wanted to learn, as long as obviously it was relevant to my project. So I really, really appreciated that from Michigan. And then, yes, and then the addiction team here was great. So that's what I was interested. My postbac was all in addiction, cocaine conditioning, and so that's what I came here for. But instead, I didn't exactly end up in addiction. Instead, I ended up in, I want to call it reward, because I work with high sugar diet, and so. But I rotated with Hudakil, in case you have any questions about that. Hudakil, Susan Shore, and then I stayed with Monica Dus. So that's my PI right now. And so what we do in the lab is that we look at how is it that the central nervous system regulates feeding behavior that then might lead to, let's say, obesity, for example, I particularly focus on the on associative learning and memory, because every day when we choose what to eat, we are making a decision, and these decisions are, the underlying of these decisions are the experiences, so the learning and memory. And so I look at how is it that diet might be affecting learning and memory, and then subsequently, how is it that this might be leading to maladaptive behaviors and then obesity. And that's it. If you have any questions, just let me know. I really like helping, so it's all good.
Thanks, guys. They were also all very modest, but I want to point out that Christina and Amanda and Thibaut, correct me, I don't know, but I know Amanda and Christina got NSF awards, so very competitive awards. And more recently, Thibaut got the Howard Hughes Medical Institute Gilliam Fellowship, which is highly competitive. And Christina more recently got the NIH DSPAN Award. For those that don't know, that's an amazing mechanism that will take you from your graduate work and support up to 4 years of postdoctoral work. So like I said, they're all superstars too, but they didn't tell you that. So I'll let you guys, and Carmen, you can chime in too. Let us know, would you rather just have an open conversation where you ask us questions, which we are more than happy to do, or you know, to minimize Zoom fatigue, or I can go through another PowerPoint if you want me to.
Oh, I don't know if the students want to have questions about toward Cristina, Amanda, or Divo, about the life at Michigan, because I hear stories that is pretty cold.
I might have to pull up the PowerPoint with the pictures of the different seasons. Yeah, and it's not cold, you just have to have the right clothes.
Well, coming from Puerto Rico, it must be a little bit adjustment, but I cannot talk much about it because I work in 2 very cold places in the United States, so I know to deal with that. But any questions from the students about the program or the highlights? What makes you different from other, programs right now that are as competitive? What is the edge that UMich has for students? For example, I would like to have a response from Cristina, Divo, Amanda, or yourself, or Audrey.
Yeah, I'll let the students talk because they're in the thick of it. And then also maybe as you guys talk, you can talk about sort of what was the hardest transition or the biggest surprise. Was it the weather or was it something else when you got here?
I think in general, so there's a lot of interaction between students and faculty within NGP. There are, seminars and there's retreats and, just maybe even seeing someone in the hallway. People will always strike up a conversation and you always try to be on your best, you put your best foot forward and always have like a plan as to how you would explain your research, whether you're talking for 2 minutes or like 10 minutes. And so that's something that at least I push myself to be able to do, is stay on top of what I'm doing, what my limitations are, and trying to improve on those things. And I think being around such like intelligent people, students and faculty, kind of push that in you. Biggest challenge, well, I'm from Southern California, similar to Puerto Rico, very warm, not as tropical, but very warm. And so it was an adjustment, but I think Shelly's right. If you wear the right number of layers and you kind of just train your body to adjust, it's not as bad. There are days where it's like, -10 and you're kind of questioning things, but I think that was probably the biggest challenge. I did move with my boyfriend, so I had some type of familial support. I would assume that if you move alone and you're really far away, that would be difficult.
Yeah, I think for me, Michigan, I was very interested in doing behavioral neuroscience, and I think from all the places that I interviewed, Michigan had just the best facilities for that. Like, I was just in complete shock when I saw the behavior room and just the access to the different resources that we could have here. And so that really excited me. And then I think having such a big program is also really great because there's just so many people like Amanda saying, that surround you at all times. So you can see, in the hallway, you'll see people from your own cohort, people from other cohorts. And I think there's a lot of interaction too between, older students and younger students. So you're always, I don't know, whenever things get tough or whatever, you can, talk to people who are older and kind of just feel better about your experience. So it's not really like an isolating program in that sense. And I think that's extremely important. And then also, which I think we've said, is the collaboration. I think it's not, I know a lot of people say that, but it's really true in Michigan. Like, there's so much collaboration between labs. I did this random experiment that then, we ended up showing the data to Dr. Flagel, and then it became a collaboration. And then, I don't know, it's just very easy to communicate with. And anytime you want to do something different, you just talk to another lab, and then, you use their resources. Obviously, like, you can replace them, but you know what I mean? It's just very easy to communicate between labs. And I think, that's kind of like the best thing for me about research. I think the challenge, I lived at home when I was in Puerto Rico, like with my family. So coming here was definitely very different. It was cold. I think the worst thing is that it's very unpredictable and that it's very long. Like, it's not even that it's that cold, it's just that it lasts forever. But then once the summer is here, it's really great. And so, I remember my first year, I saw people, like, it was one day that was like 60 degrees and people were just wearing bathing suits in their backyards, just like sunbathing. And I was like, what is wrong? And then the next year, I think I understood that when you have that one day of sun, it's like, you really got to take advantage of it. But there's a lot of, that said, there's like a lot of summer activities here. So there's like kayaking and this is summer festivals. So they really make the best out of the sunny days. But yeah, I think, again, like, just having a really strong community here with other students and the interaction with faculty definitely has made things a lot easier.
Yeah, so I think they hit all of the points very well. So I'll just talk about what's really surprised me. So honestly, I also wanted to go to Chapel Hill because they also have a really good addiction team there. But aside from the resources that Michigan had, that they had many more than Chapel Hill, there was also the fact that I wanted to see snow and I wanted to live in snow. So what surprised me is that it gets really cold here, but there's not that much snow. So I was like, what's the point of it? Like, I wanted to live in snow, but no, it gets very cold, but it doesn't snow as much. So that was a little bit upsetting at the beginning. But then as I started to know about snow and how it sticks to your car and just like how you slide everywhere and how many people just break their knees and whatnot, I'm like, okay, it's fine. It's not too bad. I'll just go way up north when I want to see a lot of snow, which is nice. The other thing that kind of really was tough on me, even though I did a post-bac at the University at South Carolina, it's always hard to be far away from family, especially us Puerto Ricans that are like family this, family that, every single like holiday, family this, and that was very, very hard for me. But the good thing about the neuroscience graduate program is their boot camp. I think the boot camp allows you to really get a connection with your peers, to the point that even now in my 4th year, we still WhatsApp all the times. And by the end of the day, you have 1,000 messages because like everybody's just like talking all the time. And then, like when you want to, just go and grab food or do whatever, they're always there. So they were very, very key in getting through the first semester and the second semester.
So I think Thibaut just jinxed us, and we're probably going to have more snow this winter than we have for the last 10 years.
then he'll have a wonderful time.
Yeah.
So there's a question from Paola, one of our Neuro-ID students. It's more of a programmatic issue. So what do you recommend for a personal statement? Meaning, what is you, Mitch, looking for in that personal statement from the standpoint of a potential student? And does graduate student read the graduate program application? Meaning, do you have graduate students in the admissions committee?
Right. So thank you for that question. So yeah, so in terms of a personal statement, my advice is first and foremost, be genuine. Don't tell us what you think we want to hear, because that we can usually tell when people are doing that. So be genuine. What got you, to this place? Why are you interested in general in pursuing a career in science? And just, sort of something, so both being genuine and something that will make you unique and stand out. So, we last year got about 330 applications. That was our biggest year yet. We have a lot of applications to go through, and so we want to be able to have something to hold on to that will allow us to remember your application. So those are my two biggest points of advice. And I don't really like giving much more than that, because it's your personal statement. So, you read that prompt and like, what comes to mind? What do you want us to know about you is really the only advice I would have. And so the two biggest things to me are being genuine and making yourself stand out, or being unique. And I can let Audrey chime in, who's been on admissions longer than I have. But then the second part, do graduate students read the graduate program application? So I am very excited that just this year we finally have implemented having graduate students join us on the admissions committee. So we will have 2 graduate student representatives joining us, and that's in the spirit of transparency, of enhancing our DEI initiatives. And I think, and just in general, I think it's going to greatly improve the process on many levels. So I think the transparency is something that, what those of us that have served on the admissions committee sort of take it for granted as transparency, we're realizing is not as transparent to students either serving on the committee or to you as applicants. So we want to be able to soon put something on our website saying, this is exactly what we're looking for. These are what we're gonna evaluate the application on. And this is what you should strive, to pay attention to when you're writing your personal statement or when you're filling out the application process. So I'm really happy with that new initiative where grad students will now, at least this year, be serving on the admissions committee, and hopefully in future years if all goes well this year. Okay. Audrey, do you have something to add?
I would just say, in addition to being genuine, which is of course really important, your personal statement is a great place to tell us about what research experiences you may have had, because we like to hear about what things you've done and what things you're really interested in. It's also a great place if there's a particular area you're particularly interested in, you might want to mention that as well. I think you've heard a lot about our addiction area. We do have lots of other areas in the program. And so if you're interested and you look at the website, because we have so many faculty, we span all the areas of neuroscience. We have strengths in sensory neuroscience, behavioral neuroscience, as you've heard, more molecular and cellular neuroscience, developmental neuroscience, translational. We have a lot of neurologists and psychiatrists that are also in the program. So there's quite a diversity there. I also noticed that someone asked about a diversity statement in the application. Mm-hmm. We do not currently have one of those, although we've been talking about whether or not we should be adding one. So I don't think we've added one yet, but there's not currently a specific diversity statement. However, you can put that in your personal statement.
Also, if you're applying through PIPS, PIPS does require a diversity statement, which I'm not sure if this is specifically for NGP, but if you're applying through PIPS as well, then I'm pretty sure they require the diversity statement. And then asking about the Toefl, I had to submit the Toefl scores.
It's not required anymore, is it, Audrey?
I don't think it's required anymore.
Yeah, I don't think so.
I didn't have to do it.
Yeah.
All right, so it was my year. Which I don't know. I think it actually helps because my score for the GRE, which I guess it's not required anymore.
No GRE.
So never mind. But yeah, never mind.
And then there's also a question, do you request a research statement or just a personal statement? And we do have both a research statement and a personal statement. Research being more, can you describe to us what your research experiences have been, in a coherent manner, and how that kind of led you to where you are, or what your general research interests are. And together, again, what we want to be able to tell from the application is we want to make sure that you sort of have a sense of what you're getting yourself into, and that this is something you want to pursue, regardless of what your long-term goals are after graduate school. But knowing that you want to go to graduate school and get your PhD in neuroscience is what we're looking for, that you have enough experience, enough research experience to make that decision, and to know that somebody at Michigan will likely be a good fit for you. But really, we want to make sure that, I can show you the slides, maybe it'll make more sense. But we want to make sure that you're prepared to allow us to help you to succeed. We don't expect you to come in being able to succeed immediately, but we want you to have the foundation so that we can help you succeed once you get here. So you can keep asking questions. I think I should pull up a slide and just go through some things quickly, because given your questions, if that's okay.
I have a question for the students. Is there any course that you decided that you saw I should have taken this course as an undergraduate before going there?
I think the neurobiology course and the neuropsychopharmacology course were the best thing I could have done, because at least for the pharmacology modules and the neurodevelopment modules, most of the information I had already learned about before. So I would definitely encourage to take whatever, like Rio Piedras, whatever you can take for neuroscience, then I would take that.
Yeah, I really think that the neurobio and the pharmacology, the neuropsychopharmacology class that Maldonado gives and that Arraraz also give are excellent. Like, I'm sorry if this bothers the admins, but I barely had to study for my exams during the first year, like barely, just the day before, just see if everything was okay, matching up, and that's it. So excellent courses.
That's awesome. Here, let me share here.
On the other end of that, while you share, I had completely psychology background, and there was not even neuroscience classes offered at my undergrad. So I took like biology courses, chemistry, physics, and psychology, and had to try to put them together. And I never had a course that did that for me. So when I came to Michigan, it all kind of melded. And I was like, oh, okay, well, I learned those two things, but never together. So you guys all have, it seems like, access to those classes, but just take as much as you can.
Awesome, thank you. Can you guys see the slides?
Yes.
Okay, so this is just showing you Ann Arbor and the beautiful seasons, the beautiful colors in the fall, the beautiful snow and football season, which we are lacking this year. I'm going to go through these really fast given the time, but Ann Arbor is essentially a great place to live, and I'm sure the students can answer your questions specifically about that. We've already highlighted how Michigan has a vast program and tons of resources, and then it's very large, very collaborative, and very collegial. And so this is, Audrey, and I think is a very valid and important point, is that we don't all study reward and behavior here. It's a huge program. It's an interdepartmental program with over 165 faculty. So this little slide was made up of, the brain in the middle is made up of junior faculty, so those pre-tenure and their research interests, and then everything around it was a PubMed search for the last 10 years or so, showing the most, sort of the most prominent articles in neuroscience, that if you put in tags for neuroscience in Michigan. So you can see there's lots of really big topics, from sleep to studying, ophthalmology and zebrafish and retina and hearing, to neuropathy, to neurodegenerative disorders. So we have a whole spectrum, and ranging from, cells to humans. Again, so I didn't say we're actually the longest standing neuroscience PhD program. We did emphasize that we're huge, but again, I think that that's actually an advantage of, as Christina was alluding to as well. And again, the reason I want to note that the reason I actually stayed here for my faculty position, even though I had other options, was because of the collaborative and collegial environment. And I just didn't get that sense at other places I had interviewed. We have a number of annual events. Again, I'm just going to go through these quickly, but if you have specific questions for the students, please chime up. So we have a welcome picnic. We have the Neuroscience Graduate Program Retreat that's student-led and organized, and faculty show up to that as well. Neuroscience Spring Symposium, where the students invite outside speakers to come give talks and hang out with us for a few days. With our large and vast alumni network of over 200 alumni, we often have a mixer and social event every other year at the Society for Neuroscience meeting. And we recently started a University of Michigan Neuroscience Conference that brings all 165 faculty and labs together to interact and present their research for a few days. Student support, you can just see here again, just kind of glance, and I want to get through these quickly, and then if you have specific questions for the students. But one thing is that there's a neuroscience graduate student organization, which allows the students to have this sort of internal governing body to make decisions and to communicate with faculty. We do tons of outreach at the University of Michigan. Yeah, was there a question? Oh, okay. Lots of outreach. We recently have grants for community wellness groups ranging from tea to soccer based on interests. We have peer-led workshops, so sort of mentorship workshops amongst students and sort of keys to success in graduate school. And like I said, recently we've implemented student liaisons on a number of our faculty committees. Okay, and so this I just want to put up there, given your questions. This is kind of the bullet points of what people often ask it in terms of what we're looking for in an applicant. And so we want students from diverse scientific, geographic, educational, racial, and ethnic backgrounds with a strong motivation to pursue neuroscience research. And so we value diversity, not just in terms of racial and ethnic backgrounds, but in terms of scientific diversity. Obviously, we didn't do a great job of representing the scientific diversity today, but like I said, we do a lot of different types of science here, so we're not going to take a class of students who all do behavioral neuroscience, so we want to be able to balance it out, you know, for those that want to focus on other things as well, of course. We want students who value community and are team players with significant research experience in a laboratory setting. So what does this mean? Well, this could mean a summer experience, or it could mean 2 years of your undergraduate experience. We have a big array of the amount of research experience incoming students have had. And again, keep in mind what I said before. We want you to have enough experience so that you know that you want to pursue a graduate degree in neuroscience. And we want you to be well prepared and have the proper prerequisites, again, that will set the foundation for us to be able to help you succeed once you get here. And so this is just a snapshot. I know that incoming or interested applicants like to see this. And so this is showing the average. Like I said, last year we had like 321 applications. Our average is around 250 over the last few years. It shows you the interviews, the offers, and the incoming trainees. You might want to see the snapshot. And so if you look at the GPA over 10 years, the range is from 2.59 to 4. So no, you don't need a 4.0. You don't even need a 3.8. You know, we take a holistic approach in reviewing our applicants. And so I want you to keep that in mind. Also, if you look, about half of our cohort last year majored in neuroscience, which means the other half did not. We recognize that a lot of places don't even have neuroscience degrees yet or offer it as an undergrad major. So we like diversity in terms of background. We take students that majored in art and music. As long as you have the prereqs that we think you need to be able to be successful in our program. And then note that about 50% of students had more than 2 years in lab, but in terms of research experience. But I want you to recognize that means that half of them did not. And so, like I said, those half that did not could have had 8-week summer training program or summer research program. So there's a big range of sort of these metrics that we don't necessarily hold you to. And then the other thing I just want to note before we end is that there's 2 routes of admission. You can apply through PIBS, a program in biomedical sciences, or through Neuroscience Direct. And that's what Christina was alluding to. So a number of our students will apply to our larger umbrella program that encompasses all biomedical sciences at Michigan Medicine, or you can apply directly to neuroscience. And once you get in via those programs, there's not much of a difference. There's some more sort of larger community-based options and possibly support through PIBs, but as neuroscience direct, you're often able to tap into those regardless. The coursework is going to be the same. The resources are more or less going to be the same. So it's a little bit confusing. And I also can't tell you whether or not you'll have a better chance of getting in through PIBs or neuroscience, because that ranges and differs from year to year based on the applicant pool. We've already been through this, your two statements, statement of purpose, personal statement, and 3 letters of recommendation. Try really hard to get a good letter from one of your primary research methods. And the deadline is December 1st. It's not rolling admissions, meaning it doesn't matter if you apply today or November 30th. And yay, no GRE, and so don't even send it. And then this is just, I don't think we need to go through this. I will send all of this so that you guys can peruse it, but I think it might be more valuable to answer last minute questions in our remaining few minutes. But essentially, you know, again, we emphasize research rotations that first year before you decide on a lab. You pretty much get your coursework done that first year, and then after that you can take electives, and that's when we start honing in on your dissertation research, helping you write grants, form dissertation committees, etc. Another thing that might make Michigan unique is that we offer a number of certificate programs. So on top of your PhD, you can get a certificate in, for example, computational neuroscience, a teaching certificate if you know you want to focus on a career in teaching, or science and public policy if you want to go into public policy. So I think that may be one of the things that's unique to us as well. And you're fully funded if you come here for graduate school. And again, this just highlights our great success in our students getting their own independent funding, which I already talked about, given the 3 students that joined us. And our large alumni group, which represents a range of careers. And I think moving forward that this tenure track faculty of around 50% is going to decrease, and all these other creative, awesome positions with a degree in neuroscience will probably emerge once we take another snapshot 10 years out soon. So with that, let's open it back up to questions in the last few minutes.
Questions?
Oh yeah, so you all 3 came through PIBs.
Yeah.
Wow, we really did not do a good job in terms of diversity amongst you guys. But we can, Audrey and I can answer questions about direct, and they can answer questions about PIBs.
Questions?
I had a question. It's regarding diversity, but it's more along the lines of what is the Latinx community around Michigan, and how integrated do you guys feel to that community?
I can start. I was the director for outreach for the Society for the Advancement of Chicanos and Native Americans in Science for 2 years. There is a huge community compared to, you know, being Michigan. And by huge, I mean pretty big. There's like 6 Puerto Ricans in just one program, and then if you go to another program, there's another 6, and then if you go to another one, there's even more that I haven't met, but like I've talked to people who know those. So there's a lot of Puerto Ricans around. There are no Puerto Ricans restaurants. I'm sorry about that. It hurts me too, at least that I know of.
Actually, they just opened one. But
they just opened one. Okay. Yeah. But for example, in SACNAS, you had a variety of Latin individuals that would gather together for like just this meetings that we would have, or get-togethers. That's better to describe it. But like SACNAS would bring in food and whatnot, and then people would just talk and mingle. There's like salsa nights, for example. So that's very nice. I actually took salsa classes for the first 2 years, and I still go and dance, and it's fun. And there's a bunch of Latin people in there. And then there's Mexican Town just next to us, which is nice to go from time to time and have some really like authentic Mexican food. Mm-hmm.
Yeah, and I think, you know, I agree with what Thibault said. And I think for me, after the hurricane, and then with the Ricky Renuncia campaign, Puerto Ricans just found each other. And I was surprised to see, you know, in a lot of, because in the neuroscience program, there's 5 Puerto Ricans. But then I just got to know a lot of other Puerto Ricans in other programs as well. So, you know, the community is here. I think a lot of it, you know, relies on you reaching out. And there's actually this huge WhatsApp that we have with a lot of Puerto Ricans in Michigan. But yeah, I agree with what Steve said.
In the final minutes, can you say something about the MD PhD program?
Yeah, so for MD PhD, you would go through the Medical Scientist Training Program, MSTP, and then from there you would be admitted to that program, but of course you would want to look through the neuroscience graduate program and make sure there'd be potential mentors of interest. And then I believe if they do it how they used to, you would rotate actually before you even start medical school, I think that first year, and then you would do the 2 years of courses, and then you would do your PhD, and then you would go back. That's how at least it used to be, but you would apply through MSTP, not through neuroscience directly. Audrey, do you have anything to add?
No, that since they just changed the medical school curriculum, they're still working out all the bugs on when the rotations are. But basically, you do the first 2 years, and I think you even take your step 1 and maybe even your step 2. Then you do your research, and then you go back in and finish more clinical rotations.
Dr. Morrow, who Christina referred to, whose lab she's in, he came through this program as an MSTP. And so if you want to email me, I'd be happy to put you in touch with him if you have specific questions. So he went through MSTP in the neuroscience program.
We also have one current MSTP student in the program and one that just went back to the clinics that we could potentially connect you with as well.
Amanda, you were going to say something? It's just got interrupted.
I was just going to echo Thibaut's statement about Mexican Town being close by. Unfortunately, there's no really good Mexican food in Ann Arbor, like the surrounding area, but like a 15-minute drive and you're there. And you get to see a little bit of the rest of Michigan as well. I think something we didn't touch on too is there's a lot of beautiful natural places in Michigan. Thibaut's taken advantage of that. There's a lot of, he's gone snowshoeing. What else have you done, Thibaut? You went to Picture Rock.
Yeah, I went to the up north to Picture Rocks. I also went to Sandals. Sorry, that's my dog. I went to just a lot of kayaking. There's snowboarding, obviously skiing. There's just a bunch of lakes around. There's recreational fishing. Yeah, there's a lot of hiking. Yeah, there's a lot of things to do here.
Yeah. So I've been here about 25 years, and during COVID, I've discovered even more parks and trails and ways to enjoy the outdoors.
One of the other things is, I think Shelly mentioned we have community groups now, and one of our community groups is an outdoor group, and they go on hikes in the area, and they also went camping up on Sleeping Bear Dunes, I think. So there's lots of things to do.
Sounds wonderful. It really does. I really do. I want to.
It is. I'm biased, but I think it is.
It highlights the quite, how you say, well, I guess the different opportunities for growth in so many ways that UMich has to offer. And then we really thank you all for being here. I guess we're 4:30, so we're going to wrap it up. And I don't know if Dr. Garcia wanted to say something to finish. No lo veo por ahí. Yo creo que se fue. I think he left. Maybe the internet or something. But for myself, thank you. Thank you so much, Shelly, for doing this. I know we wanted to do it through the regular seminar, but this went out wonderful. And then we have more opportunity to have the students to engage and talk about University of Michigan, which is a wonderful program. And to my former students, besito, amore, thank you so much. You make us proud, and for me in particular. And Amanda, you're not from my crowd, but that doesn't matter. You are part of us, and most certainly, I wish you the best in your career, and obviously to Cristina y Diego. And so we thank you again for your time and your consideration with us. And I guess we'll see each other, and you will see applications for sure coming from our cohort. So thank you so much for being here.
Awesome.
And I'll talk to you guys later.
Okay, thank you so much for having us, and thank you, Mish Crew, very much for your time. Thanks, everyone. Take care. Stay well.
Bye. Take
Bye.
care.